further ado, here's Adam Ierymenko with Evolvable Living Machines.
All right.
Today I will be presenting on what I refer to as evolvable instruction set virtual machines. I'm not sure if there's a canonical name for this technology, so this is what I call them. Some other people call them artificial life virtual machines, but I think that's a little too vague. So the basic characteristics of these is that they have machine languages at the low level that are evolvable. Now, what does it mean for something to be evolvable? First of all, to be evolvable, you have to be able to withstand small changes without completely falling apart. There has to be more than one way that the system can work, and the system has to be able to be mutated without crashing, without anything catastrophic happening. Not all systems are evolvable. I'm sure a lot of people have heard it's an example that's frequently brought up by skeptics of evolutionary theory, is the idea of, if you have a clock and you go into a clock and you randomly move components around, it's not like the clock is going to gain new function, the clock is just going to break. Well, the reason that the clock breaks is that the clock is brittle, which is what we could call the opposite of evolvability, or what another term for evolvability that's sort of similar that evolutionary biologists use as robustness. Now, for something to be evolvable,
sorry, I got a question.
Go ahead.
Sorry, already. Isn't one of the reasons you can do mutations is because a lot of the stuff just doesn't do much at all?
There is a certain reality to that. As a general rule, most evolvable systems have what you would call 20% neutral mutations, which means that on average about 20% of the mutations that could occur to the system have no effect on the function of the system at all, or negligible effect.
Okay.
But generally, in an evolvable system, you have kind of what mathematicians call a bathtub curve, where some of the mutations, about 20%, have no effect at all. The middle part, maybe about 50%, have very small effects, very incremental effects. And then the tail of the curve, which goes upward, you have some mutations that have very large effects. And of those, the vast majority of them are fatal. But there's a very small fraction of that tail that is not fatal and that actually adds to the functionality.
Right, thanks.
So when we're talking about evolvable instruction sets, we're talking about instruction sets that have the characteristic of robustness. Now, almost all computer languages that have been designed by human beings are brittle. They are not robust. If you open an executable file in, say, Microsoft Windows, or if you open a C or Java source code file, and you go in and you randomly change a few bytes of the file, almost all the time the result will be that the file, if it's a source file, it's not going to compile, or if it's an executable file, you're going to run it, and at some point in using the program, it's just going to crash. The reason for that is
Have you done the experiment?
Have I done the experiment? I've had hard drives that develop bad sectors, and that's basically doing the experiment.
It also depends how many comments there are.
Yeah. Oh yeah. Comments don't count. Comments are inert. I mean, actual program code. But the reason that they are that way is because we designed them to be that way. When human beings designed engineered systems, systems that are designed to do one specific task, we usually want them to do only what we tell them to do. We don't want them to do anything else. For example, when you go to your ATM and you withdraw money, your bank does not want the ATM because of some random bit mutation on, say, the hard drive inside the ATM to decide to calculate your checking account balance differently. The bank would much rather have the ATM simply crash and say out of order, and then you go to a different ATM. This is common in a lot of engineered systems, actually, not just computers, where we will actually design the system to break cleanly and in an informative kind of way when it changes outside of its design specifications. This also helps us to write software. It helps us to debug software because when the program does something that's outside of its specifications, it just crashes, and then we can trap that and we can look at what was happening. So generally, we don't want our programming languages or our low-level instruction sets that are executed by computers to have the characteristics that would make them evolvable. Now, what's kind of interesting is that in the early days of computer science, there was actually a bit of a synergy between computer science, well, at that time it wasn't quite differentiated from mathematics, and the subject of biology. Most people don't know this, but two of the earliest computer scientists, one of them being Alan Turing, who is widely considered to be the founder of the field, and then John von Neumann, who's considered to be one of the people who helped to mature the field, were both interested in biology. Alan Turing actually, I guess you might say, moonlighted as a biologist. Go ahead.
You just have to talk louder. You have to project to get back here.
All right. Alan Turing, I guess you might say, he kind of moonlighted as a biologist. He did a little bit of work in biology in what's called Fibonacci phylotaxis, where he actually worked out the mathematics of the developmental structures that are formed by a number of organisms, in particular flowers and plants and organisms that have geometric structures. John von Neumann, on the other hand, was very interested in the mechanics of self-replication. He designed a form of computer system called a cellular automaton, which a lot of people have probably heard of. And he started asking sort of philosophical questions about what elements would a system like a cellular automaton have to have in order to be able to make copies of itself. And one of the things that John von Neumann did that was kind of interesting was he correctly predicted many years before biology actually discovered how information flowed in a cell. He predicted that you would have what he called a tape, which was like the DNA, and then he predicted that there would be some sort of translation layer that would translate from that tape to an end product, which in biology is called the phenotype. So he more or less predicted the information flow of living, self-replicating cells before that was actually discovered by the field of biology. So in the early days of computer science, there was a connection, a crossing of interest between biology and computer science. After that, computer science mostly went off in the direction of trying to solve problems that were very well defined. Engineering problems, problems in domains like business data processing, cryptography, that sort of thing. However, there was always a tremendous amount of speculation in computer science about could you use biological principles, in particular evolutionary principles, to actually design things or create things in the computer science domain. There was some early experimentation in the 1960s, going back, I think maybe even a little farther than that, on the subject of genetic algorithms, and that's a very large subject in and of itself that I don't really have time to go into here in detail. But one important point I wanted to touch on was that by the 1980s, around the early to middle 1980s, there were people using genetic algorithms and genetic programming, which are forms of machine learning algorithms that use evolutionary principles to actually synthesize software programs to solve specific well-defined problems. And the idea of a genetic algorithm is usually you run a simulation where you have a big population of programs, you explicitly assign them fitness based on some kind of fitness test that you come up with. And then the fittest survive generation after generation, and at the end of the process you take out an end result, that is the program that you've synthesized. Now, one interesting sort of side point I wanted to make is that before the early 1980s, at least as far as I know, and there might be elements of the history that I'm not aware of, there wasn't really that much success in actually using evolutionary principles to produce programs. Once genetic programming really kind of took off as an accepted method in the mid to late 80s, everybody kind of recognized that this actually could work. you actually could use evolutionary principles to synthesize computer software. Now, genetic programming usually did not operate directly on the computer program that was being evolved. Usually you would have some sort of meta representation, like a program tree or a parse tree or something like this. There may have been some work leading up to this in genetic programming, but the first evolvable instruction set machine that has an artificial life sort of bent to it that I'm aware of is Tierra, which was created by a man by the name of Thomas Ray. And Thomas Ray was actually a biologist, and he learned about a system called Core War. Core War was a game that was actually created in the early days of computer science, where it was essentially competitive virus writing. Basically, what people would do is they would write computer programs in a simple language called red code, and then they would run them in this virtual machine, and the object was to write programs that could self-replicate, that could make many, many copies of themselves, and could also kill other people's programs. So the objective of Core War was, by the end of the game, your program would be the only program running in this virtual machine. Tom Ray knew about Core War, and there had been a couple of attempts to get Core War to evolve by adding mutations to the system. And he thought, well, the reason that Core War doesn't evolve very well is that the instruction set is too brittle. So he took what he knew about genetics and the structure of the DNA code and the requirements of evolution, and he designed an instruction set that wasn't brittle. He essentially took the brittleness out of the instruction set. And what he came up with was the Tierra instruction set, which, when placed in a Core War type environment, actually does demonstrate evolution. Now, if any of you, I'm sure some of you are familiar with machine codes and instruction sets, the main things that Tom Ray did in Tierra is, first of all, he got rid of specific jump targets. In computer instruction sets, you'll have, of course, branching instructions. Everyone's familiar in high-level languages with ifs and loops and those sorts of structures. And in a computer instruction set, that's what these structures translate down into when a compiler compiles them. Now, typically in a computer instruction set for a processor like the x86, you will have an instruction like jump, and then there will be a number afterwards, which will specify where the instruction will jump to. So you'll have a jump instruction which says something like, jump forward 550 bytes. Now, the problem with that is if you mutate that, let's say a bit mutation occurs and changes it to jump forward 10,550 bytes, that's going to be a very, very large change in the structure and function of the program. So as a result of that, you're going to have, instead of having that nice bathtub curve where, say, 20% of mutations are neutral, 50% of mutations are minor, and then 20%, 25% of mutations are major, you're going to have a situation where almost all mutations are going to have very significant effects. And that tends to prevent things from evolving very effectively. So that's one thing he did. Another thing that he did is he made sure that all of the instructions were the same length. By making sure that all the instructions were the same length, that meant that if you inserted or removed segments of the instruction stream, you didn't shift the reading frame of the instructions forward or backwards. The reading frame was always the same. So that again eliminated another form of catastrophic mutation. If you go to the web page for this particular talk, there's actually a link to an example of the Tierra instruction set. And you can see what the instruction set actually looked like, and you can see some of the details of how he accomplished this. But when you put this instruction set into a Core War type environment, into an environment where the programs can self-replicate and they can interact with each other, Tom Ray actually observed some evolution and even some simple ecological behavior, where you would have, for example, programs that would use each other to help each other self-replicate, sort of like parasitism. And then you would have the evolution of mechanisms to resist that. So you have the evolution of parasite resistance. So we saw some simple ecological phenomena occur. Now I'm going to go over a couple of other examples of evolvable instruction set virtual machines, just to give you an idea of what these can be used for. One of the most significant uses of virtual machines like this right now is in the study of evolution, in particular in performing what you might call impossible experiments. Because when you're studying evolutionary biology with real living things grow at a relatively slow rate. I think the doubling time for the fastest bacteria that are capable of self-replicating is something like 45 minutes to an hour.
20.
20?
20 minutes.
20 minutes for it's E. coli, isn't it?
The phages that infect them, 7 minutes.
7 minutes.
So
the fastest evolution you can possibly generate right now in a biological system is a generation about every 7 minutes. In a computer, you can have a generation about every, say, 1 or 2 seconds or less. So, first of all, you can run much longer experiments. Second of all, you can, because you're the computer programmer, you're running it in your own virtual machine, you can basically play God. You can set up the conditions any way you want, and you can also log absolutely every event that ever happens inside the simulation. So, for example, you can log every mutation that ever happens to any cell, or you can log every offspring you can tie to its parent, so you can develop, exact trees that describe how the system evolves over time. And that allows you to take hypotheses that in the real world are only really observable indirectly, and actually design experiments that directly test those hypotheses. So one of the uses that has emerged right now for this type of system is theoretical evolutionary biology research, and this has actually been used to generate papers that have been published in major science journals like Science, Nature, and Journal of Molecular Evolution, and journals like this. So this is actually
wrote the paper itself.
What?
What? It wrote the papers itself.
No, it generated the research that helped the people to write the papers. Now, one of the evolvable instruction set virtual machines that I've created is called Nanopond. And at the end of this presentation, if we still have time, I'm going to show a video of that, which it's kind of neat. It's a 15-minute long video. I'll kind of forward through it and just show you the most significant parts. But it shows Nanopond evolving for about 8 hours of computer runtime. Nanopond is written in C, and it's only about 332 lines of C code. The reason that I wrote this is because I had people emailing me a lot, asking me questions about how do I write these sorts of systems. And so I decided to write one that was very, very small, so that computer programmers that were fluent in C could just look at the source code and figure it out. And it was actually quite successful. A lot of people all over the world have modified it and have added their own extensions and done their own experiments with it. And it's also been used in a couple of computer science courses as teaching material to illustrate how to write a simple virtual machine, and also just because it's neat and the students like to play with it. So if you go to the Gray Thumb website, if you click on projects, you can download the source code to this and binaries and see screenshots and whatnot. Now, something else I've been looking into, going further with uses of this type of system, is using evolvable instruction set virtual machines as a machine learning platform. Now, this would be kind of like genetic programming, except instead of selecting for a single individual at the end of the genetic programming run, instead what you would be doing is you would take a problem and you would make that problem a feature of the environment. And then the entire population of cells would essentially adapt to that problem. So the end result, instead of being a single program, would be a population of them that had adapted to perform the task that you wanted them to perform. And there's a system that I've created that is not available for download yet, but it's called Ouroboros, and it's designed to do just that. And so far, it has done, I would say, medium difficulty pattern recognition. The way you have it do pattern recognition is it can differentiate between patterns by you make every pixel in the image that you want it to differentiate what's called a resource in the environment. And the cells in the environment are able to examine the resource, and they're also able to interact with it by what's called metabolizing the resource. And when they metabolize the resource, they provide a category, and if that category is correct, they receive a reward, which gives them greater access to the computer CPU. So this is what it's done so far, is images of about this level of difficulty. You have shapes with sort of a fuzzy background that I'm currently working on using it to do quantification for cell assays, where let's say you have an assay which is a photo of a bunch of cells on a microscope slide, being able to look at that and quantify how many cells are in that photo. It's one of the things I'm experimenting with using that for. So to summarize where we are right now, ignore that, I took a slide out. But what we have instruction sets that are like computer machine code instruction sets that can suffer mutations without catastrophic effects. We have virtual machines that run those that are able to generate populations of programs that can exhibit ecological behavior and evolution. We are using that as a tool to do theoretical evolutionary biology research, and we are also have the beginning of systems that use that as a machine learning technology. And where do I think it's going? Well, one place it's kind of already going is that we can finally test concepts from evolution and ecology that human beings couldn't really directly observe because we don't live long enough. Obviously, we can't watch a system evolve through several ages of evolutionary time, but we can in a simulation. And then the second thing where I think all of this is leading, not only this, but also genetic programming, which Martin is going to talk about next time, is toward the day when people say, remember when people wrote software by hand. So that's it for the presentation part, and I'm going to show you a video of one of these systems evolving over a period of time so you can see what this actually looks like, because a picture actually tells you a lot more than listening to someone stand up here and talk about it. Now what you're looking at right here is a blank screen. And the reason that the screen is blank is that at the beginning of the simulation, the virtual machine is empty. There's nothing in here. There are no programs running in the virtual machine. Now, what the virtual machine is doing at this point, and you can't really see this because this isn't actually shown, is that the virtual machine is randomly generating lots of programs. Basically, it's taking a random number generator, it's dumping the output of that random number generator into cell programs, and then it's executing it. Now, 99.99999% of these don't do anything interesting, but the tiny percentage that do begin to make copies of themselves. So if we start the video, you will see dots starting to appear on the screen. What those dots are is they're cases where a randomly generated program has been able to make a copy of itself.
How does it know?
How does it know? When a program makes a copy of itself, the virtual machine attaches a number called a generation. Every time a copy is made, it increments the generation, and it only shows pixels that have a generation greater than 0. So the vast majority of the time, you don't get what's called a stable self-replicator. But when you do, you get these formations where you see a little colony of cells starting to form. And right here you can see a rather interesting one. These colonies of cells, once they start to form, once you have a stable self-replicating cycle going, they will start to colonize the entire virtual machine.
Why do they like to go in straight lines?
Well, that's actually the simplest thing you can do. If you look at the instruction set, And by the way, you can download Nanopond and look at the instruction set from the Graythumb website. If you look at the instruction set, about the simplest self-replicating program you can possibly write is one that simply copies to the first adjacent cell. And if you do that over and over again, you're going to get a straight line. Now, if you do something a little bit more elaborate, you're going to get patterns like this. And this one, obvious.
The default neighbor is your left neighbor?
Yeah, the left neighbor is the default neighbor that they all start with. So if they're not changing which way they're facing, then they're always going to be self-replicating to the left. So you'll get straight lines. These obviously are changing which way they're facing before they self-replicate. Now,
Different colors are different.
different colors are they're color-coded based on a checksum of the genome program. So different colors indicate different genome programs. These aren't sexually reproducing in any sense. This is much more basic than that.
This is G-rated.
Yeah, this is G-rated. This is not. Although on the website I did actually call this A-life porn, but technically it's not because it's asexual. So it's pretty soft porn. If it were the reason I was going to say that is that in biology, there's not really a hard and fast concept of a species when you're talking about asexual organisms. Everything just kind of bleeds, especially bacteria in very simple cells. So in this case, you know, you can roughly say that a color corresponds to a species, but it's really a much better way to classify them, at least in a rough sense, it's just a watch. And when you see certain behaviors emerge, I kind of name.
Where are the mutations coming from? Is it just a replication error or something?
There is the virtual machine, as it executes the programs, randomly introduces variations into the state of the state machine that's doing the execution. So because the programs actually copy themselves, when random noise is introduced into the state of the virtual machine, it causes them to occasionally make copy errors. So that's the origin of the mutations in the system. Go ahead.
Are more similar colors more genetically similar?
Yes. The colors are, although the algorithm I used to do that is a little, might not be perfect. So really the only thing I'd say for certain is that different colors are different genomes. So like this purple line is all the exact same program. Now, these ones here that start to develop, these are very interesting because see how they're kind of a rainbow? What that means is as these are self-replicating, they're actually changing their own genomes. So these are actually polymorphic. You can see as they replicate, they generate different colors. Now I'm going to step forward. Well, actually I don't need to. I was going to say I'm going to step forward. Oh darn, you won't be able to see it. I've watched this video before, and there's something that happens down in this area.
It's happening right now. It's projecting.
Oh yeah. Oh yeah. You can see it. See this red that's starting to form down there? Oh yeah. That's a new behavior that's emerged that I call the blob. And the blob will eventually take over most of the screen. And what's neat about this video is this video is about 8 hours of actual runtime, but it's condensed down into 15 minutes. So you're basically watching a fast motion video, and you get to see it evolve through a couple of different ages, kind of like Earth has been through, you know, the age of the fish and the age of the dinosaurs and the age of the mammals. In this case, we have the age of the initial radiation, followed by the age of the blobs, followed by the age of the diffuse blobs. So you can see these guys starting to take over, and you can see there's been another mutation down here that gives rise to an even denser species. Go ahead and let that down. There's going to be another mutation up here that will give rise to something that actually starts eating the center out of this very dense sort of growth that's happening up there. And
So how do they compete for resources? I mean, what's...
well, there are two limiting resources in this virtual machine. One of them actually, oh, see, there you go. See how it's got kind of a growth in the middle? But, sorry, to answer your question, there's two limiting resources in this virtual machine. One of them is what I call energy, which energy corresponds roughly to access to the computer's microprocessor. And if a cell runs out of energy, it ceases to exist. The second limiting resource, of course, is space. The virtual machine is only so big. So once it gets completely colonized, space starts to become a commodity. So now you have two resources that are being competed over. And when the programs run, every time an instruction executes, that costs 1 unit of energy. So in order for the program to execute and do anything, it has to consume energy in order to do this.
Where do you get the energy from?
There's a background process that randomly places energy into the space at a certain rate. So there's sort of a background energy that's always flowing in, and the cells can then find that energy and
Is it distributed uniformly through the space?
yeah, it's distributed uniformly. You can, if you want, you can edit the source code and change it so it's not distributed uniformly. You can make it distributed,
The tropics.
say, in a certain area. You could have the tropics, and you could have the Antarctica, where there's almost no energy. Now you can see the rainbow guys have their little colony here. And you can see a couple of other quote-unquote species that are kind of colonizing. This guy is kind of interesting. Remember the species that kind of grew in the middle here that started to eat it from the inside? You can see that. It's kind of yellow. It's right here, and it's kind of coexisting with this, but it's kind of broken the blob apart.
What happens with the boundary between orange and purple?
I believe they kind of run into each other. We can kind of forward it a little bit. We don't have to go through the whole 15 minutes. But they kind of run into each other. Now you can see the orange and the purple kind of run into each other, but they are kind of closing out all of the other species. So eventually they come to dominate space. And we're down to this holdout here, but as you can see, another species has actually emerged right here. So we have this holdout being closed out, sort of crowded out by these guys, which are very successful.
So why don't we see any mixing?
Why don't we see any mixing? There's two reasons for that, actually. Well, you do see a little bit, like on the frontier, you see a little bit of crossover. One reason is that
But it's asexual, right?
well, it's asexual. Another reason is
I'm
that...
simply talking about spatial. Why don't they
spatial
penetrate?
mixing. The cells in this simulation actually can't move. They can only copy each other.
Right.
So there's no way for a cell to kind of swim. So that's one reason. There's a guy on the internet that actually added that. And when you add it, you get much more of a well-stirred reactor kind of effect where the cells kind of intermingle with each other quite a bit. It doesn't look quite as neat as this because in this you get to see the species as very well-defined areas. The other reason for that is that it's only a two-dimensional grid. So it's impossible for two cells to be on top of each other. So when one cell is very successful and takes over a region, you don't see very much competition in that region until something comes along like these guys that is more aggressive and can actually fight it out. So you can see this colony is actually getting larger. So it's actually starting to...
The red and the purple, they have similar rates of
they seem
reproduction.
to. They seem to. If you look at this, they seem to have a very similar behavior. In fact, I haven't done a rigorous analysis of the data from this particular run, but it looks very much to me, just visually, that these two species are actually doing more or less the same thing, and this could very well be a neutral mutation.
They came out of the same
They
sort of
came out of the
cluster,
same
so-
cluster, and the color difference could very easily be a neutral mutation.
How many instructions are
In the programs?
yeah.
These guys, I think the average for this running substrate was a couple hundred. So the mean genome size for this particular run was a couple hundred instructions.
Do you know how many instructions were inert?
No, that's actually kind of hard to figure out. Back when we were having smaller gray thumb meetings, I ran a simulation called Arcus, which is another one that I wrote that was prior to Nanopod. And Arcus can do what I call random genesis too, where you randomly generate cells and eventually one of them can self-replicate. We took one of the genomes of one of the randomly generated cells and we printed it out, and then we sat there for about, what was it, like 45 minutes or so, and we couldn't figure out how this thing was making a copy of itself. And it was only about 50 or 60 instructions, but we were tracing through it and we were like, well, let's see, it's reading from its genome and then it's XORing it and then it's shifting it left twice and then it's doing a branch instruction based on, it was doing something incredibly bizarre, but somehow it was self-replicating. So it's actually quite difficult. not quite on the order of reverse engineering actual living organisms, but it's quite difficult to take these guys apart because evolution does not have the same design biases that human beings do. And when evolution designs programs, it will create things that are quite bizarre and very different from the way a human being would design them.
Carl Sims found the same thing with his.
Yeah, the crawling fig guys. He'd have, some of them would move by swinging, and
He said that his programs were like 80 to 100 instructions, and that most of them seemed to be just junk that didn't do anything.
oh yeah, they do all sorts of strange things. So you can see there's another down here that is different from this, although this could be a neutral mutation. I'm not sure. And these are highly aggressive too. And we've gone down to just one little surviving colony of the
Might be extinct.
polymorphic species there. So you're seeing some extinction go on, and you're seeing a takeover by one type, and then you're seeing the emergence of a new type. See, these here are the mammals, and these are the dinosaurs. And we can go forward. Yeah, we can go forward and watch the triumph of the mammals here. You can see that.
Is each program at a fixed size so that it can be represented? Its survivability or whatever is represented by one pixel, or can they ever grow and expand so that they're
Oh no,
represented
the
by
programs are
large
very
pixel grids, sort of like it glues itself to become larger and larger?
actually multi. What?
They're all in a single cell? Or
They're all in a single cell. This is a very simple simulator. As I said, it's only 322 lines of C, so there's not a tremendous amount of really rich ecological interaction
That
that's
includes
possible.
that makes the movie too?
Actually, yes and no. I mean, there's
GPU stuff there?
it outputs PNG files for every frame, and then I use something to encode it. But yeah, that includes what can output the image files to make the movie.
But in theory, multiple pixels could have mutual programs.
Yes, they
The system
could.
create something that
Oh yeah, it's not theoretically impossible for something that exhibits multicellularity to evolve in here. I've just never seen a really striking example of it so far.
That took a long time.
Yeah. Oh, it took a long time on Earth. I mean, for the first, billion years, you would have, I mean, if you come to Earth during the first billion years or so of life, you would have visited and you would have seen endless mats of bacteria. And then you would have come back 200 million years later and there still would have been endless mats of bacteria. It took a very long time for anything apparently interesting to happen. So this is only 8 hours of computer runtime. I don't know what would happen if you ran this for 6 months because I haven't done it.
How fast is your computer?
It's a 2 gigahertz Athlon. So this is this 15-minute movie is 8 hours on a 2 gigahertz Athlon. So if I had a computer I could lock in a closet and a really huge hard drive, I could run it for weeks and weeks. What?
What size grid are you on?
This is a 640 by 480 grid. And the maximum size of each cell is 512 instructions. So I can't do math in my head, but if you multiply that, that tells you the maximum memory size of this. It's a couple hundred megs, I think. Now you can see that this sort of phenotype, where they're much more diffuse and they're much more aggressive, is now out-competing this phenotype, which is a little denser and a little slower. At least that's how it looks on the movie.
the upper yellow is different from the lower yellow.
This? Yeah, that's a slightly different shade.
It's more of a neon.
Yeah, more neon. I think so.
You talked about tracking the species based on, keeping a history of everything as it
Yeah.
evolves.
This
Did
doesn't
you do that for?
Oh, no, that would fill up my hard drive.
It would be
So,
just very interesting to know, just
It would.
blues
It
versus
would.
the greens,
You
seeing
could
where
do
they
that. You
split
could-
off, and
I mean, it would also execute a lot slower because it would be constantly dumping tremendous amounts of information to the hard drive.
well, the fact
which-
that you have sort of a finite number of species here,
Right.
you could keep some sort of rudimentary data about.
The combinatorial explosion basically.
Yeah.
Or
Yeah.
just save the top 10.
Yeah,
Yeah.
if you
Sunday, blues versus green.
But this is why we can't keep tracking the evolution itself. It's like there's just
Oh,
too much
it's
stuff out.
very, very hard to do. One of the things that's interesting about these artificial life systems is that It's one of the only things that I know of, other than some of the applications in biotechnology like genomics, where if you gave me a building the size of a football field that was full of nothing but racks and racks and racks of computers, I can think of experiments I could do that would saturate all of that. And would still, and at the end of the experiment, I'd still be going, if we had a computer twice this big, we could do something twice this large. It's one of the few applications for computers that we have left that is really making us want faster and bigger computers.
All the computations physics.
Along with role-playing games, those are.
A bunch of calculations in physics are like that.
Oh yeah, physics, scientific computing in general, I think. But most people don't do scientific computing.
How much
Just scientists.
do you think there's an interaction between your energy field?
Energy
the
field?
The random insertion of energy,
Uh-huh.
which allows the instructions to run,
Yeah.
and the diffuse
So close
pattern
to
that we
extinction.
see within the block.
That's an interesting point, because what we could be seeing actually here in the pattern itself is we could be seeing a kind of a bizarre, kind of multi-dimensional plot of the behavior of the Mersenne twistor random number generator. It would be interesting to
I
hook
think
this.
red has been made extinct. Oh, there.
Yeah. Well, the red is almost extinct. It's going to go extinct very soon. This is live action video. You can watch. You can watch the struggle for life. See, isn't this a lot less boring than my presentation?
Better than the World Cup.
Right.
But they're not actually competing to reproduce into the cells.
Actually, they do to some extent. They can also kill each other. I mean, they can interact in that cells can take energy from each other. They can step on each other's spaces, that sort of thing. So like I said, both space and energy are commodities in this environment. Although space doesn't really become a commodity until the entire environment is saturated. But the patterns that you see, although the random number generator I use here is a random number generator called Mersenne twistor, and it has a period of something like, I actually remember this number, I think it's something like 2 to the 17,790 something huge like that. So basically you'd have to run this for about 2 or 3 million years to get a repetition in the random number generator. Nevertheless, pseudo-random number generators do sometimes generate patterns at higher dimensions. So it's an interesting point. It'd be interesting to hook this up to a hardware random number generator that samples.
Well, that's not my point. The point
Oh,
is
it is?
the curdiness of it.
Uh-huh.
You said that if a creature runs out of energy,
It dies.
It dies.
Yeah.
And so you have a sort of critical mass.
Oh
Creatures
yeah.
that are able to reproduce,
Uh-huh.
that are being fed by the
Yeah.
energy source.
So basically what you could be seeing here is very small, radiation followed by extinction cycles.
Exactly.
And they
And
reproduce very quickly.
If you turned up the energy, you might get denser.
Yeah.
Turn up the sun.
Yeah.
Yeah, but you saw really dense ones show up early.
Yeah, but they might have depleted their environment, and then that's why they might have become less dense. Actually, if we rewind, we can see. See, there's very dense ones right here, but they might be depleting their environment. See how they're leaving kind of an empty space? They might be
Because the
depleting.
energy was already left there from...
Yeah, the energy was already left there, so they're depleting their environment. That's a good point. And then finally, these guys reach an equilibrium, and what it might be is that these guys are more energy intensive. They might be using more energy during the course of their execution. So they, even though they seem to be more successful, they seem to be able to displace these guys. They might be using more energy to do so. So as a result, you get a more diffuse pattern. You get a pattern like this rather than a denser pattern like this. That's very true. Go ahead.
So we definitely can see, you can classify that there were 2 major things, then there were 3 major things, and there were 5 major things, and so forth. You could actually count what
Yeah.
the big players.
At least visually, and that's not really scientifically rigorous. It's basically a human sitting there and going,
Well,
"Ooh, look."
histogramically, I think you
Yeah,
do
yeah.
a fairly good job.
Yeah.
So if you made a chart of the frequency of their appearance, what do you think it's going to do? In other words, are they getting better and better, and they're going to eventually saturate your instruction set so nothing better can be done, or do you expect it to them to get better?
If you graph these, I've actually done a thing where every so many steps it outputs a complete image of the entire substrate into a file, and then what I can do with that file is I can run a program on it that first goes through it, selects only the active parts of every program using a couple of heuristics, and then I run it through a tight data compression algorithm like VZIP2. Now, what data compression algorithms do is they eliminate redundancy. So if you count the number of bits that comes out of the other end of your data compression algorithm, that'll give you a rough kind of relative measure of how much non-redundant information is in the space. And if you do that, you get a graph that kind of looks like this. You have the initial radiation of many, many different types, and then it plateaus, and then it dives down, and then it kind of levels off. And this is actually a behavior that you see in many, many of these systems, and it's actually exactly what evolutionary theory would predict. Because what we have here is we have an environment that is perfectly uniform. This environment is in and of itself featureless. There's no mountains here, there's no valleys, there's no weather, there's nothing really that's cyclic about the environment. The environment is totally flat, totally featureless, and therefore what's going to happen is evolutionary theory predicts that in a featureless, flat, stable environment, the key word being stable, you're going to have, once you get a highly successful organism, you're going to have what's called stabilizing selection. So that's eventually what you end up with here, is you end up with stabilizing selection, and you end up with a system that continues basically like this. Now, like I said, I have only let this run for 8 hours and then I stopped it. As far as I know, it's impossible to prove, given a system with this many dimensions, this many possible degrees of freedom, that is in fact the endpoint of the system. I seem to think that would require solving the halting problem, which Turing proved is impossible, because this is a Turing complete system. So you can't say with absolute certainty that at some point in the future it wouldn't spike up again and maybe settle back down again. And it would be interesting to run one of these systems for months and months. However, I know that the people that work on Avita at Michigan State, if you go to the website for this presentation, there's a link to that lab. They have actually done experiments on their own cluster where they have executed Avita for, I believe, several months, and it does show that sort of behavior where you'll get a long tail and it just doesn't do anything for a long time. Now, one thing that I had found, that I thought of that was quite interesting was, there's a couple things I'd like to try with this. One is I'd like to try putting weather into the system, where you have some kind of cyclic variability, maybe like a warm season and a cool season, and what that would translate into is variations in how much energy is being put into the space. Another thing that would be neat would be to add a lot of random sort of maze-like walls into the space to add some texture to it.
What would a wall mean?
A wall would just mean something that's impassable, or something that can only be passed if you pass a certain energy threshold, something like that.
So that wouldn't be a regular feature in a cell, that would be another...
No, that would be a special feature of the environment. Oh. And then another idea that I had that, hey Peter. Another idea that I had was, it was actually an idea that I had when I was at Artificial Life 10, and there was a lot of discussion of this plateau phenomenon that you see in these systems, and there was a little bit of discussion of is there anything that we could do to get continuous evolutionary change. One idea that I had that I haven't tried yet that I would like to, or if any of you guys want to beat me to it, you're welcome to, is to take this two-dimensional grid that this inhabits and create kind of a double grid where you have two grids on top of each other. The top grid would be occupied by the simulation that you see here, and then the bottom grid would be occupied by a class 4 cellular automaton, such as Conway's Game of Life. And the idea would be that these cells that are executing on the top would interact with the bottom grid, and that would be the environment they would execute against. And so that would give them an environment that was that they could change, and that would change in sort of, I guess you would say, non-deterministic, but also non-random sort of ways. And so the idea is you could have the development of self-sustaining ecologies where things would mess with the underlying CA and do neat things to their environment. And I haven't tried that yet. If somebody wants to hack Nanopond and stick a class 4 cellular automaton like Conway's Game of Life in there, and have that be the environment, and then see what happens, just reference me in the paper.
Is there competition for cells, and is there one program's trying to copy itself with one cell to the left, and another one's trying to copy itself with one cell to the right, and they're both fighting over the same cell?
You can actually get that.
Does one of them win if it has a higher generation number or something? Or what?
The algorithm for winning and losing those sorts of battles is that the cells have their first codon, I believe it is. Codon is a biology term. The first number in the instruction stream is used as what's called a binding site. And when two cells interact, it measures the number of bits that are in common between those two numbers, and it uses that to do a probabilistic assignment of which one wins the conflict. So you can actually have the evolution of different sorts of binding sites where cells of the same species will be allowed to interact with each other freely, but a cell that has a different binding site won't. And that's actually something I use in Ouroboros and a couple of other systems too. Any more questions?
Something that's kind of different from all the
Yeah.
artificially, made virtual machines like that, and actual biology is that the programs that are being evolved seem kind of uniform, more or less. Maybe I'm wrong, but in biology, like in a real DNA segment, there are places where you can, where a mutation could be much more significant. Like for example, if you mutate the gene which regulates how many times cells will copy themselves versus the actual structure of a certain one protein.
In a computer program, you can have the same phenomenon. I mean, in these programs, you could have. Restart that.
Actually, you're right.
In these programs, you can have parts of the program code that are sort of pivotal, in the sense that if this changes, it's going to radically alter its behavior. And then you can have some parts that are kind of inert. One thing you have in evolved computer programs is you'll get formations like, take a number, add something to it, subtract it, add something to it, and subtract it. Well, that doesn't do anything, but it's there. And then if you mutate something in there, it might have a very small effect, but if you mutate two things in there, it might have a very large effect. I don't have a video, unfortunately, of Ouroboros because, actually I do, but it's on the internet. I have to get to a website to view it. But Ouroboros is n-dimensional, where n is a number that you assign when you create the substrate. And most of the experiments I've done training it to do things like pattern recognition, I've used numbers of n like 5 and 6. And so you have a 5 or 6 dimensional space. And I have yet to figure out how to graph that. So, there's no video of that right now. I guess you could do something with OpenGL and crazy vector mechanics, but I haven't been that how would I put it? Bored. Lazy. Bored. Yeah, I haven't been that bored yet.
Along the same lines as that question, you talked a lot about brittleness and instruction sets.
Yeah.
And why don't we see, instead of seeing 2 or 3 species dominating the entire field, why don't we see a whole rainbow of inert mutations? Things which are basically the same, but which are having these sort of neutral mutations spreading across the entire field.
Well, you do kind of to some extent. I mean, you can see.
But later on in the video.
Yeah, later on. I think that what happens later on is that the substrate kind of gets conquered by a couple of species that represent fitness maxima, and mutations from those fitness maxima tend to be detrimental, and so detrimental cells kind of get crowded out by the already existing. So
even when you eliminate brittleness on the lowest level, you can still get into situations where the higher level, the more sophisticated genomes become brittle themselves because they become so
Yeah.
wedged into a certain.
And you have the same thing.
things.
The same thing in biology, although to a lesser extent. Biology still has better evolvability characteristics than we have been able to generate so far in an instruction set. And the reason is that in biology you're talking about real chemistry, which is an analog system. And there's just an analog system like that has characteristics that we have found very difficult to replicate in their essence in a program, which is basically a logic tree. However, we've done well enough, as you can see, that evolution actually does happen. But the second point I'd make, by the way, I didn't really introduce my background. I actually studied biology in college. I didn't study computer science. So that's kind of where all of this came from. But another thing that you have as a result of just the dynamics of evolution is when you get an environment, and you get this in nature too, that is dominated by a single fitness peak. If the environment is uniform, evolution favors maintenance of that fitness peak. And that's called stabilizing selection. So evolution will tend to maintain whatever the current fitness peak is, unless something comes along that happens to be at a higher fitness peak.
I got a question, well, kind of a thesis, a little bit of a thesis.
Okay.
Between brittleness and like parallelism. Because when I look at this, I see all these things kind of going in parallel and try and figure things out, and some of them do good, some of them do worse. Now, when you design a machine that's going to give you one answer, it seems to me like you're designing a machine that's brittle. And the reason that comes up actually is in a real application. I happen to have diabetes, okay? And there are people out there who are trying to build a machine that will tell 24 hours a day what your glucose level is. And as soon as they figure that out, my disease is, basically super well managed. They haven't done that. You looked at biology and you say, what do the cells do? Well, there are these islets of Langerhans, and each one of them measures the glucose level, and each one of them does a crappy-ass job. And it's hard to tell an engineer, build a meter that does a really lousy job. But the point is, they build 10 million of them. And when you have the 10 million of them, then on average, they kick ass.
This is called Google.
Well,
Google builds computers as cheaply as they can to fail as often as they can.
Yeah.
So I'm thinking that, based on looking at those cells, looking at your video, a real characteristic of robustness is just asking the question 100,000, a million times, kind of sloppy, don't care about the nut and bolts details, but then you extract from that the average behavior, and it eventually gets to a real efficient answer.
Yeah. The reason I went to this slide when you were talking about that is when it does the pattern recognition, when Ouroboros evolves to do this, what you'll have like, and I haven't spent the time to do a really rigorous analysis of this, but I have taken a look. And what you get is about 10 different species of cells in here, all of which are using very poor rules of thumb to try to categorize. Like, for example, if this pixel is below this color and if the red, green, and blue are equal, then it's this. Well, that might only work, 20% of the time. But if you have 10 different overlapping different species that are doing 10 different things that work 20% of the time, and if you're taking a cumulative result, eventually, in the case of this particular example, it went to 100% efficiency at pattern recognition. So that's an example of what you're talking about when you use large numbers of basically large numbers of poorly effective things. You can get very effective results.
Yeah, and the point is it's
and
robust.
robustness. Because if you go in and you kill one of the cells, it just dies.
Yeah.
But the whole system keeps going. Whereas in a clock, every part is absolutely integral. Go ahead.
So I have a kind of a question all the way back to the beginning.
Okay.
We call these evolvable instruction sets, I think.
Yeah, evolvable instruction set virtual machines is what
But
we
the
call them.
instruction set is fixed. So the instruction set supports...
I see what you're saying. Yeah, I see what you're saying. The instruction set itself is fixed. Evolvable program virtual machines? Well, in this case, evolvable instruction set, you're talking about a characteristic of the instruction set, not the instruction set itself.
Evolvable instruction sequence virtual machines.
That's pretty good. That's pretty good.
Change the acronym.
And you don't even have to change the acronym. Yeah.
But what does ISVM mean?
ISVM? Nothing. That
Does
is.
anyone else use your acronym? Or is that?
I've asked before what the canonical, proper name is for these things, and I've never gotten the same answer from more than one group of people. So, therefore, I
But
kind
the
of
one
just
group
came
is
up with
pretty consistent.
one group will be pretty consistent. So I came up with my own name for them that seemed to make sense.
So is there any serious effort into looking at one instruction set being better than another instruction set for this one?
Yeah, that was something I actually brought up in a discussion at A-Life 10, and I'm working on a system right now which I nicknamed it Elemental because it's designed to be the most minimal instruction set possible. It's a virtual machine that executes only NAND instructions. Now, NAND. What'd you say?
I asked what you said.
Oh yeah, NAND. And if you know anything about digital design, you know that there's two logic gates that it is possible to build any digital circuit out of. Those are NAND and NOR, and NAND is the more commonly used of the two. So what I did is I created a virtual machine that I already proved it's Turing complete by using it to do a bunch of operations that would be present in a Turing complete language. And it's a virtual machine that executes only NAND instructions. So there's only one instruction, and it's NAND A to B to C. Now, what I want to do with that, and I haven't, this project has been kind of languishing since I've been working on the Ouroboros project, and I have a day job and 50 other things I'm doing.
A, B, and C, a memory location?
Yeah, like 50 other things I'm doing.